Thank you for joining us for Metabolon's half-hour webinar series, where our topic today is Secret Weapon, Interrogating Host-Pathogen Interactions with Precision Metabolomics. Your presenter for today is Dr. Jeffrey Feld. Jeff received his PhD in biophysical chemistry from the University of California, Berkeley, where he used structural biology to shed light on how anthrax toxin acts like a Trojan horse to infect host cells. While a postdoc at Lawrence Livermore National Lab, his team discovered and characterized novel potential virulence factors from tularemia, the rabbit fever, with an eye towards developing countermeasures against bioterrorist threat. Jeff joined Metabolon in 2017, and as a study director, he's responsible for analyzing and interpreting large metabolomics data for industry clients with a focus on applied markets, which include human and animal health and nutrition, consumer goods, and skin care. I'll be turning the presentation over to Dr. Feld now.
Thank you for that introduction, and I'm glad to be here to talk to you today about how metabolomics can support infectious disease research and hopefully impact new discoveries. So ever since Alexander Fleming discovered our first antibiotic, penicillin, in 1928, there's been a constant battle between the development of novel antimicrobial therapies and bacterial mechanisms of antimicrobial resistance, such that resistance to newly developed antibiotics always occurs, as you can see in this top graphic, sometimes even before introduction of the new antibiotic into the population. In addition, only a handful of low-hanging fruit strategies for bacterial killing and growth mitigation have been exploited. And in the past few years, the development and approval of antibiotics, particularly novel forms that are not just tinkering with previous methods, has generally been on the decline. A number of reasons for this decline exist, which include scientific, economic, and regulatory bottlenecks. The compressed story is that the short time of treatment and cure nature of these compounds means they're not as economically as appealing as chronic treatments, and the regulatory approval mechanisms are geared towards disease state approval rather than bug class, which ties into those economic barriers. And at the same time, antimicrobial resistance, or AMR, is alarmingly on the rise. A recent report on antimicrobial resistance published in the UK estimated that annual deaths worldwide from 6 major pathogens would rise from the at least 700,000 that was observed in 2014 to potentially 10 million by 2050, a number that is greater than current deaths from cancer and diabetes combined. This, of course, is an estimate based on current trends and mitigation strategies. The take-home message remains that unless new strategies are undertaken, we'll be paying dearly for the price. So what we've learned in previous webinars is that metabolites, which are the small molecules that represent the end result of everything else, can be represented as, or thought of as a surrogate for a phenotype, so that everything else includes genomics, diet, and xenobiotics, as well as our microbiomes, that other organ that lives inside our gut and in our skin. And for more information on resident microbiota, I urge you to look at our webinar from November of 2017. But what we learned from that webinar is that changes to the microbiome can result in an altered metabolic homeostasis, which takes into account changes from the host and also these metabolites that are the currency, the interaction between the host and the microbiome. And this results in an altered phenotype that can be expressed as disease or changes in behavior. For the sake of this webinar, we're going to focus on pathogens. So those are microorganisms that make us sick. And in a lot of the same mechanisms, the interactions between the host and the pathogen are brokered by these small metabolites or immunomodulatory metabolites. And on the host side are mostly integrated by immune cells. So this infection results in an altered metabolic homeostasis, so this altered phenotype that we call the infection. So perturbations in metabolic homeostasis caused by these pathogenic microorganisms can be measured as changes in metabolites in a technology that we call metabolomics. So I'm not really going to go into details on this particular slide, but I wanted to show you this really nice figure from a recent review from Christopher Sussetti's lab at the UMass Medical School, published in the last couple of years. And Professor Sussetti has done some work with Metabolon, but really shows this integrated nature of the host and the bacteria, the infectious bacterium, within this closed metabolic system. So basically, what this boils down to is that the host and the bacteria are competing over the same micronutrients, so carbon sources, amino acids, nucleic acids, as well as micronutrients. And the difference is between host and bacterial metabolism, these organisms can sense the presence of their adversary, adjust metabolism accordingly, and try to gain a competitive advantage. So as an example, bacteria can ferment a lot of these macronutrients, and this results in molecules that are recognized as foreign by the host and results in a host response that's mostly given in the form of inflammation. So the host can recognize these molecules and elicit a response. And so the scarcity of nutrient resources in this closed system also drives competition. So basically each set, the host and the bacteria are trying to put the other side into a metabolic state that's not really their preferred state. And this can really, so basically you can consider infection to be this metabolic arms race where changes in metabolism drive infection. So I wanted to give you a couple examples of kind of how this competition arises. So amino acids in particular are central in the metabolic arms race. Not only are they represent those scarce resources, but they also act as ammunition in the fight. So for instance, tryptophan is an essential amino acid that activated T cells convert to kynurenines via the action of an enzyme IDO, indoleamine 2,3 deoxygenase. And while kynurenines themselves have immunomodulatory aspects, the action of depleting tryptophan essentially removes this molecule, this essential molecule, from the metabolic space, and so bacteria have to compensate. For instance, the pathogen Francisella tularensis that causes tularemia has developed a de novo tryptophan synthesis cascade so that it can still produce the tryptophan it needs even when activated T cells are depleting the stores. In a different mechanism, Chlamydia trichomonas senses IDO-mediated tryptophan depletion and enters this non-replicating persistent state, or quiescent state. So by using host metabolic cues as a signal for entering this quiescence, chlamydia is able to cause long-term persistent infections even in the face of ongoing immune responses such as the depletion of tryptophan. And once that tryptophan levels come back up to normal, it can go back into its more pathogenic state. So another amino acid that's important, plays a central role in infection metabolism is arginine. So this central metabolite is used by inducible nitric oxide synthase, so INOS, of the host to synthesize nitric oxide, which inhibits bacterial respiration. And also, arginine can be converted by arginase 1 into an ornithine, which fuels polyamines, which also plays roles in later stages of infection, including wound healing. Now, Staphylococcus aureus has developed a mechanism where to avoid this nitric oxide toxicity by expressing an inducible lactate dehydrogenase, which promotes bacterial survival by maintaining redox balance. So in this case, it can switch to a fermentation state and avoid the toxicity insults by nitric oxide. And in a different mechanism, chlamydia expresses an ornithine decarboxylase, which deprives INOS of the essential amino acid arginine and converts it into polyamines. And this, otherwise, instead of being affected by nitric oxide, chlamydia is able to convert the arginine into polyamines. And actually, along this route, new clinical isolates of Staphylococcus have recently acquired what's called an arginine catabolic mobile element, or ACME cassette, which enables them to directly resist polyamine toxicity because they can catabolize those polyamines that's going to affect them later during the infection. So altogether, we can see these mechanisms by which the immune system and the pathogens that are attacking have developed mechanisms to sequester and use these scarce metabolites and also use them towards their competitive advantage. So looking at the immune system side more in particular, innate immunity recognition of specific microbial metabolites, so these are pathogen-associated molecular patterns, allows the immune system to recognize that it's under attack. So a lot of these molecules are prokaryotic in nature. They don't exist in the host cell, the host organism, and they include the nucleotide derivatives such as cyclic dinucleotides that can control biofilm formation, for example, and also the organic acids that are produced by fermentation. Infection-induced activation also alters the metabolism of host cells, and this changes the bioavailability of important metabolites for the pathogen. For instance, there are molecules that are secreted that sequester divalent cations, and actually it's even been shown that microbiome-related divalent cation sequestration is important. For instance, E. coli and other enterobacteria secrete the inhibitors that are analogs for Pseudomonas metallophore molecules, blocking their reuptake. And additionally, cells and macrophages can trap pathogens in intercellular compartments, so basically limiting their nutrient access. And then there's also metabolic pathway inhibitors, including reactive oxygen species and nitric oxide that we've discussed, that are general inhibitors of bacterial replication. And I wanted to show you this one pretty cool recent discovery of how activated macrophages produce this molecule, or an enzyme called cis-iconotate decarboxylase, which converts the TCA cycle intermediate cis-iconotate into itoconate. And itoconate has specific inhibitor activity of a bacterial-specific enzyme, isocitrate lyase, that controls the rate-limiting step for glyoxylate formation. So this glyoxylate cycle is unique to bacteria. Mammalian cells don't really possess glyoxylate pathway. So in this mechanism, activated immune cells can block this important pathway for bacteria that it uses often in the face of oxidative stress or when it needs to replenish central carbon metabolism by blocking a specific prokaryotic enzyme. So now I want to go over ways, a particular way that we've developed at Metabolon for interrogating these changes in metabolites. So I've shown you a couple different examples of why metabolites are important during infection, and we've developed over the last 15 years this integrated technology approach to metabolomics that is geared towards building knowledge. When we call this precision metabolomics, this technology. It's more than just a set of mass spectrometers. It starts first with good study design. So we have scientists that can help investigators develop a good study design with controls, some consulting on sample prep issues, and making sure that we have enough statistical power. Once those samples arrive at Metabolon, they're run on all four of our platforms, these methods that are designed to provide optimal coverage of the metabolome. And this generates huge amounts of data, on the order of about 150,000 ion features in the mass spectrometer per normal sample. But instead of an ion-centric approach, we've developed a chemocentric approach consisting of sophisticated software and an extensive library of metabolite standards and quality assurance and quality control methods that permit us to eliminate noise and reduce these data down to a list of metabolites that are actually present in the sample. And then from there, using bioinformatics and statistical approaches, our team of biochemists and study directors, like myself, are tasked with interpreting the data in terms of the scientific questions of interest so that we can give you actionable information based on the data and the study design. And I just wanted to demonstrate quickly kind of how this works. So this graphic here represents the atlas of the metabolome, where the arrows indicate curated biochemical pathways, and each blue dot represents a metabolite that we typically see in most sample types. So what we do, we get your biological samples, which could be biological fluids of any type, cell pellets, cell supernatants, tissues. We've done way more than these type of samples, but these typically are what we receive from customers. And we screen the entire metabolome across your samples to find key changes or differences that are important, keeping in mind the goals of your study and questions that you want answered. So you see these changes between various study sets, so your control and your experiment, or within experimental groups, and we see these changes in metabolites, and we can interpret those in terms of biochemical pathways, in terms of the questions you want answered, and hopefully provide key insight and biomarkers that will aid you in your research moving forward. So for the rest of the webinar, I want to give you a few examples of how Metabolon's technology has informed host-pathogen interaction studies. So the first study here actually came from Christopher Sussetti's lab that we talked about earlier, the review that I used to demonstrate some examples. And his lab was interested in the mechanism of cholesterol catabolism and Mycobacterium tuberculosis, or MTB. So MTB is an intraphagosomal pathogen that must rely on the host cell for all of its nutrient requirements. So it was speculated that it may actually catabolize lipid bilayer components for these carbon sources, since it's locked within this lipid bilayer within the macrophage cell. So the Sussetti lab had previously shown that cholesterol degradation was necessary for the growth and survival of MTB during chronic infection. But it wasn't really clear whether this catabolism of cholesterol contributes to central metabolic requirements, and if they can understand on a more mechanistic level of how the cholesterol is catabolized, they could develop potentially new methods for looking for therapies against MTB. So what they did was they grew tuberculosis on two different carbon sources, glycerol and cholesterol, and sent us at Metabolon the cell pellets, and we performed metabolomic profiling on these cells. And so this profiling revealed that MTB grown in cholesterol showed increased levels of the metabolites malate, fumarate, succinate, and shown here methyl citrate. And all these are collectively components of the methyl citrate cycle, MCC. So this cycle, if you see down here in the bottom right, converts propionyl-CoA to pyruvate for insertion into central carbon metabolism. And the investigators showed that the cholesterol, that actually more than 80% of the propionyl-CoA that they measured in the study was derived directly from cholesterol. So you could say that potentially the MCC is this pathway to get cholesterol into central carbon metabolism. So to test this hypothesis that MCC is this important pathway, they tested the growth of MTB in macrophages. So in this hypothesis refinement box, you can see that's the left strain of MTB, and they produced a mutant lacking two of the key enzymes in the MCC pathway. So that's PRPD and PRPC. And this mutant was the growth of this mutant within macrophages was severely inhibited by lacking essentially lacking the MCC pathway. And as a nice positive control, the researchers also supplemented this knockout strain with vitamin B12, which allows MTB to bypass the MCC pathway by directly converting propionyl-CoA to succinyl-CoA via methylmalonyl-CoA, which is a methylmalonyl pathway, MPP. And since under these conditions, tuberculosis can't de novo synthesize vitamin B12, this would demonstrate that in lacking of the MCC pathway, it could still deposit cholesterol carbon into central carbon metabolism. And sure enough, it can, just like wild type. And so this really locks down that this mechanism of MTB catabolism of cholesterol, locking it down in this MCC pathway of delivering propionyl-CoA into central carbon metabolism. So this provides a new potential target for M. tuberculosis, because we have these enzymes that are specific only for bacterium, and we can potentially develop novel drugs or treatments that can target this specific pathway. And interestingly enough, a group, Perhar et al., were intrigued by these results and reasoned that statin therapy, which as we know lowers cholesterol in the host or in humans, may provide protective therapy against MTB infection. And sure enough, in a macrophage model, they derived macrophages from patients who are taking statins. These cells were actually more resistant to MTB infection compared to those of healthy donors. And then using a mouse model where they treated mice with statins and then subsequently infected them with MTB, showed also that the statin therapy increased host protection. So potential path forward here for translation into humans. targeting cholesterol catabolism and in tuberculosis. Excuse me, I went to the last slide there. I'm going to show you my cards. Here we go. So the next study here came out of Jim Collins' lab at MIT, and they were interested in the antibiotic susceptibility of Pseudomonas. So Pseudomonas aeruginosa is a primary pathogen associated with cystic fibrosis lung infections. And antibiotic treatment, largely with aminoglycosides like tobramycin, of patients who are infected with Pseudomonas is associated with reduced infection burden, but not actually eradication of the bug. And this antibiotic failure could be attributed to one of two possible outcomes, or two possible reasons. One being a genetic resistance, such that possibly acquiring and expressing resistance elements, or a phenotypic tolerance. And so in this case, it would be adopting some kind of physiological state in responses to the local environment, so an environment-dependent mechanism of antibiotic resistance rather than a genotypic failure. And it turns out that aminoglycosides, so they require proton motive force to cross the bacterial cells and block protein expression at the prokaryotic ribosome. And so it's the bug itself, so Pseudomonas in this case, can adjust its metabolism, say by becoming more anaerobic and less cellular respiration in order to reduce drug uptake. And that was sort of the hypothesis here, was to determine the biochemical basis of this antibiotic susceptibility. So before we jump into the approach, let me give you a little bit of background on how this worked. So the Pseudomonas susceptibility to tobramycin depends heavily on the growth phase. So while exponential phase cells are quite susceptible to tobramycin, it's the stationary phase cells that display the antibiotic tolerance. So it's, again, this phenotypic rather than genetic mechanism for altered sensitivity. So based on this, the investigators hypothesized that potentially they could stimulate metabolically. We okay? Okay. They could stimulate metabolically these tolerant cells to tobramycin by adding different central carbon metabolites and seeing if they could stimulate different activity. So on the left graphic, you can see they added a supplemented stationary phase tobramycin treated Pseudomonas with various central carbon metabolites, and then tested their ability to protect or enhance the activity of tobramycin compared to control. And what they found was that metabolites associated with central carbon utilization pathways were actually altered the susceptibility depending on which branch of carbon metabolism they're looking at. In particular, the lower TCA cycles, such as that include fumarate, increases Pseudomonas susceptibility towards tobramycin, whereas the glyoxylate that we talked about earlier in the bacterial-specific glyoxylate shunt actually increases tolerance towards susceptibility. So based on this, the susceptibility to tobramycin definitely depends on central carbon metabolism, and this is where the metabolomics comes in. So investigators grew Pseudomonas on one of three different carbon sources, either a control or supplemented with fumarate or glyoxylate in the stationary phase, and grew up the cells, sent us the cell pellets, and we performed global metabolomics on the stationary phase cells. And so you can see glyoxylate is in blue, fumarate in red, and control in black. So compared to controls, glyoxylate supplemented cells showed significantly increased levels of the lower TCA cycle intermediates, malate and fumarate, citrate, and alpha-ketoglutarate, while acetyl-CoA was significantly depleted, to the point that only one of the five cell pellets actually showed acetyl-CoA levels that were above the lower limit of detection. So these results indicate that glyoxylate could potentially be inhibiting alpha-ketoglutarate dehydrogenase, which would result in an increase in the alpha-ketoglutarate citrate levels, while also depleting acetyl-CoA through the glyoxylate pathway and fueling malate synthase. So that would increase the levels of malate and fumarate, and thus bypassing the lower TCA cycle. And what this has an effect is now that we're bypassing the lower TCA cycle, we're bypassing the creation of reducing equivalents that would be used in oxidative phosphorylation. So essentially, by bypassing the lower TCA cycle, the glyoxylate addition is tuning down oxidative phosphorylation. At the same time, the fumarate-treated cells, most of the TCA cycle intermediates were largely unchanged compared to controls, except for acetyl-CoA, which was also significantly depleted. And so this implies that the TCA cycle was stimulated by fumarate, depleting acetyl-CoA and resulting in increased cellular respiration. And if you have an increased cellular respiration, you should be enhancing susceptibility to tobramycin versus the glyoxylate effect of reduced cellular respiration, which would increase its tolerance to tobramycin. So to test this hypothesis, the researchers measured respiratory activity measured by oxygen consumption rate, OCR, and sure enough, fumarate supplementation increased oxidative respiration, while glyoxylate supplementation decreased respiration compared to control. And interestingly, if co-supplementation of glyoxylate and fumarate results in reduced respiration. So this glyoxylate reduction in cellular respiration seems to be the dominant effect over the fumarate stimulation. And to further hammer home that point, the researchers showed that respiratory activity is directly related to tobramycin susceptibility. So we're looking at here percentage of survival on the y-axis and addition of glyoxylate to tobramycin supplemented and fumarate supplemented Pseudomonas cells grown in the stationary phase. And you can see that glyoxylate addition protects these cells from sensitivity to tobramycin in a concentration-dependent manner, as well as a time-dependent manner, whereas this last graph here shows the amount of time passing between the time difference between the addition of glyoxylate. So up to 30 minutes, glyoxylate can rescue this activity that fumarate enhances, and then it starts to lose this activity after 30 minutes. So both in a dose and time-dependent manner, glyoxylate seems to be the dominant factor in tolerance to the antibiotics. So as a take-home message here, the researchers showed that the susceptibility of Pseudomonas to tobramycin antibiotic treatment can actually be tuned by altering the central carbon metabolism of the pathogen using various carbon source supplementation. And so, in effect, the reductions in respiration effects seem to dominate the drug uptake effect. So there's more than just the potential of drug uptake by this proton motive force. There's something else at work here as well, where increase in cellular respiration seems to be the dominant effect, as shown by glyoxylate fumarate co-addition. And metabolomics really played a key role in showing how the TCA cycle is this central player in tuning the susceptibility and providing key information on the mechanism. So fumarate may actually represent, in this case with Pseudomonas, a useful adjuvant with tobramycin treatment of Pseudomonas infections and cystic fibrosis. And this really could be a potentially interesting theme or paradigm in looking at current antibiotic treatments and looking at ways we can supplement central carbon metabolism to enhance the activity of those antibiotics. Now, of course, this is going to be bug specific. What works for Pseudomonas might not work for E. coli, but it really gives us a route forward in using our current antibiotic strategies and supplementing them with something potentially easy like central carbon metabolism, and using metabolomics as a key technology to integrate that. So now the last study I wanted to share with you came from the Faber lab at Thomas Jefferson University, and here we're not looking at bacteria but looking at a virus, rabies, and the effects of rabies vaccine on protecting against post-exposure treatment, or PET. So rabies, as we know, is an infectious disease of the central nervous system, CNS, that caused by RABV, the rabies virus, and more than 70,000 people worldwide die annually from rabies, and several million more undergo post-exposure prophylaxis. So this is a large burden on our healthcare. And currently, there's no approved treatment for humans who develop clinical rabies, and this is mostly attributed to features of the infection that enable RAB-V to evade immune clearance from the central nervous system. So kind of a promising approach to this is generating attenuated RAB-V vaccines that can be used as effective post-exposure treatment in animal models. And so the one we're going to be focusing on here is Trigas, which is a recombinant RAB-V attenuated vaccine that has been shown to be highly immunogenic and non-pathogenic in normal and immunocompromised mice. In designing this study, the investigators had previously conducted a transcriptomic study that revealed that there were genes associated with adaptive immunity that may be important for this Trigas mediated viral clearance. But they understood that study was lacking in a real phenotypic understanding, so they went for metabolomics to see if they could understand through a brain metabolic changes whether they can understand better this mechanism of Trigas viral clearance. So for this approach, these mice were infected with rabies virus, and 4 hours post-infection were treated with a mock or a vaccine, and then at the given day indicated 4, 6, or 8 days post-infection, the mice were sacrificed and their brain tissues were submitted for metabolome profiling here at Metabolon. And so among many of the brain biochemical changes that were observed, one of the most striking was this dramatic increase in corticosterone levels observed in the mock-treated mice and after 8 days. Which is, an important time point within the accumulation of rabies virus within the brain. And these levels this increase was largely eliminated with the Trigas vaccine treatment. So to validate this study and understand if the same increase in corticosterone levels was also present in serum as well as brain, the researchers developed an ELISA assay to track serum corticosterone levels during infection, and found that, as you can see in red, increases in serum corticosterone levels were significant only in the mock group over the course of infection, whereas those treated in the blue group that was treated with Trigas, this increase was largely reduced. And so we know that corticosterone, as a glucocorticoid, is a stress hormone secreted by the adrenal cortex and regulated by the hypothalamic pituitary axis, or HPA. And previous studies have revealed that the rabies virus, and as well as other viruses, can activate the HPA. So this molecule corticosterone that has potent anti-inflammatory properties likely reduces the immune response and immune cell differentiation and proliferation activities that would be important for eliciting an immune response in the central nervous system and aiding in viral clearance. So to test this hypothesis, that levels of corticosterone are being increased by the virus, and this is potentiating its ability to evade the immune system, they treated RAB-V infected mice with daily metapronone, which is an inhibitor of corticosterone synthesis. And these mice that were treated with metaperidone showed significant reductions in serum cortisone levels using their ELISA assay, as you can see here in red. As you'd expect, the mock treatment increased significantly during the course of the infection, whereas the blue cortisone inhibitor treated mice showed no increase in corticosterone levels. And interestingly enough, starting at 10 days, the metaperidone treated mice showed significant improvements both in clinical signs, so that's measured as a reduction here you can see in blue in the clinical score, as well as increased survivorship, significant increased survivorship. So essentially blocking the production of corticosterone within these mice was enough to rescue a lot of them from the RAB-V-induced death, so improving clinical scores and survivorship, simply by just inhibiting corticosterone. So to summarize, metabolomics in this case revealed a number of metabolites showing that brain biochemistry was different between the Rabies infected mice that had received post-exposure treatment and those that hadn't. But most particularly was this increase in corticosterone levels. And corticosterone, as a glucocorticoid, blocks inflammatory processes that promote immune cell activation, migration, and infiltration in the central nervous system. So production of corticosterone as a result of this HPA activation could impair the innate immune response. And it's been shown for several virus infections, including Rabies, activate the HPA. So by reducing corticosterone levels with an inhibitor, this resulted in improved clinical and survival outcomes. And this, the researchers attributed this to blocking its immune suppressing activity. Since you need these pro-inflammatory mechanisms, example, antibody-mediated protection are needed for this protection against Rabies. So by blocking anti-inflammatory corticosterone production, you're allowing the immune system to eventually infiltrate the CNS and clear the virus. So with that, I just want to summarize the webinar here. So that to a pathogen, infection can be considered a problem of microbial ecology, whereas it needs to adapt to this new and exciting environment of the host and needs to accumulate scarce resources that in effect is in competition with the host. Well, at the same time, the immune system uses metabolism to sense changes in its local metabolome that are induced specifically by the pathogen, either microbe-specific metabolites like PAMPs or just changes to the local metabolome that are indicative of a pathogenic insult. And a lot of its tools and defenses that the immune system possesses are geared towards forcing the pathogen out of its metabolic comfort zone, where it's either sequestering resources or forcing it out of central carb metabolism, amino acid metabolism in ways that eventually will weaken and allow weaken the bug and allow the immune system to kill it. And hence, measuring and interpreting these changes in metabolites with metabolomics can provide valuable information towards realizing new biomarkers for not only identifying disease, but also tracking infection, or also, as we've shown here, discovering novel microbial mitigation strategies.
Thank you. We are running kind of close on time, so we'll probably have time for about one question. And just a reminder that if you've submitted questions during your registration or if you've submitted questions online and we don't get around to answering those here, we will reply as soon as possible offline.
So the question is, can metabolomics measure antibiotics in addition to endogenous molecules? The answer to that is yes. So on our platform, we have a number of common antibiotics that are in use in the marketplace. So we can readily measure those. We can also look at some breakdown catabolic products of some antibiotics. And if you're a researcher who is developing an antibiotic treatment, a novel treatment, we can work with you to put that molecule on the platform and measure it and make sure that it's working the way you think it is, and do so, in a manner where we're not telling your competitors. Yeah, so in addition, the endogenous molecules, we've talked about a little bit in this presentation, like kynurenine and tryptophan, arginine. We measure those. We measure downstream catabolites of many of these. That some of them are bacterial specific, some of them are host specific. So yeah, we have pretty nice coverage of I think most things that you'd be interested in terms of the host microbe interaction.
Thank you again for attending our webinar. In case you'd like to share the replay or listen again, the recording will be available soon on our website at www.metabolon.com.